Use of multi-photon fluorescence lifetime imaging is an assay technique for carcinogenesis and pharmacological efficacy. Quick outline of what's going to the webinar for today. First of all, we're going to have a review of technical aspects of FLIM and implementation in Bruker FM multi-photon systems. That's going to be done by Mohammed Nazir, who's one of our applications engineers. And then we're really pleased today to have Dr. Melissa Scala from Vanderbilt University with us to present some really interesting data from her laboratory. Melissa's general research area is development and translation of optical imaging until May 22nd. I won't be able to check this voicemail very frequently. If you'd like to reach me, please contact me through email at jeff.stuckey@bruker-nano.com. Thank you. Sorry about that. Melissa's general research area is development and translation of optical imaging and spectroscopy techniques to quantify changes in optical biomarkers at the cellular, metabolic, blood oxygenation and concentration, microvessel network structure, perfusion, and collagen content. And her major focus is on cancer and cardiovascular disease. And at the end, then we'll have questions. You can type in questions at any time during the presentation, and then at the end of the presentation, we'll field the questions for you. So, and then also when the webinar is completely done, there'll be an opportunity for you to fill out a brief survey for us to let us know what you thought of the webinar today. So at this point, I'd like to turn the presentation over to Mohammed.
All right, thank you, Jeff. So I'm going to start with an overview of fluorescence lifetime imaging as a technique by itself, and then how we integrate that into our Bruker fluorescence microscopy systems, mainly multi-photon systems. Lifetime imaging is based on the fluorescence property called lifetime. What we basically do is create a 2D map of lifetimes, and then that's what gives us our fluorescence lifetime image. And then I'm going to quickly go over fluorescence. I guess most of you are actually familiar with that. It is the emission of a photon from a molecule in singlet excited state. The picture you see on the right here is the Jablonski diagram. Most of you have seen it elsewhere. A molecule is actually in its electronic ground state right about here, and then it receives some energy, like a certain amount of energy.
That phone length recorded. To re-record, press 1. To delete, press star D. If finished, press pound. For help, press star H. To have system wait, press star W. If finished, please hang up. Or, to disconnect IP office, press star X. Please make entry soon or be disconnected. To approve, press pound. To record from here, press 1. To play back, press 2, 3.
Okay, I think we were having some technical glitch, but I'll keep carrying on.
Goodbye.
So we were talking about fluorescence. So here's where the molecule would receive this certain amount of energy. It will go to the absorption event. It will go into its excited state. The excited state is not stable, so the molecule would like to go back to the ground state. It will emit a photon after a certain time. And that emission, if it happens on a nanosecond time scale, would be fluorescence. If it happens on a longer time scale, it will be phosphorescence. The molecule can also go back to the ground state through some non-radiative process, in which way we won't see any emission of photons. When fluorescence occurs, there are certain properties that we can actually measure. The most common one, or the most familiar one, is the intensity, which is the number of photons per unit area, and that is what we do when that is what we measure with our conventional fluorescence microscopes or laser scanning microscopes with multi-photon imaging or confocal imaging. Another property that we measure is the wavelength of the photon. This is a measure of the energy of the photon, and we use different detectors or spectral detection to measure that. and a couple of other properties are lifetimes and the polarization state of the molecule. And we're going to talk about lifetime here. Quickly, the lifetime is a measure of how long a molecule stays in the excited state. So in simplest terms, it will be the difference in time between the absorption event here and the fluorescence event here. But it's actually not that simple because fluorescence is a probabilistic process. So if you do it only once, you're unlikely to get the right lifetime value. You have to repeat the process several times and build a histogram and then extract the lifetime value from that histogram. Lifetime has certain properties that are very exciting. One of them is that it is independent of concentration. So like whenever we do intensity imaging with multiphoton scopes, because intensity depends on concentration, it's kind of hard to quantify, but lifetime is independent of concentration, so that's a very nice property. The other property is that the lifetime can be altered by the microenvironment that the molecule resides in. And actually Dr. Scala will talk more about this and give you experimental data and results about that. But essentially the lifetime can depend on the pH, temperature, presence of oxygen, or proximity to other molecules with certain spectral characteristics. So how do we measure lifetimes? We can measure lifetime with frequency domain techniques as well as time domain techniques. The time domain techniques are more popular with laser scanning microscopes, and we use a technique called time correlated single photon counting that can be easily used with scanning microscopes. And we will talk about that in a little bit, but before that I would like to show you this exponential decay graph. So like I said a little while ago that to get the lifetime you have to have like an exponential decay, and this is what this shows. For example, at time t is equal to 0 here, you have a large population of molecules, and all these molecules are in their excited state. And these are all similar molecules, they're in the same environment. If you start measuring the fluorescence intensity over time and then plot that intensity over time, what you would get is a single exponential decay curve. What that means is that at about 5 nanoseconds, which is the lifetime for this curve or this kind of molecule, at 5 nanoseconds the intensity would drop down to about 37% of the value. And this is the equation for a single exponential decay curve. If you have more than one type of molecule, or if you have a molecule in 2 different states, you would actually get a double exponential decay here. So moving on to TCFPC, the time correlated single photon counting technique that we use with data scanning microscopes. In this technique, we actually measure the time of arrival of every single photon with respect to the most recent excitation pulse. In this technique, the key is that you keep your excitation power so low that you only get a single photon per excitation period. So in this picture here, what you see are these red arrows that indicate the excitation pulses from a pulsed laser. Let's say this laser is an 80 megahertz laser, so then these red arrows are 12.5 nanoseconds apart. And then the green arrows indicate the arrival of photons. When the photons arrive, we can measure the time between the arrival of photon and the most recent excitation pulse to get a certain value for this particular fluorescence. And once we have a number of these, once we have a number of these events, we can actually build a histogram. So what we do is we sample the single photon emissions for a large number of excitation pulses, and this helps us construct this histogram. So what we have done is that this 12.5 nanoseconds here has been divided into 6 time intervals. So let's say one interval, the first time in is like 0 to 2 nanoseconds, and the next one is 2 to 4, 4 to 6, and so on. And then as we measure the photons, we put them in their respective time bins. So the bins here contain the number of photons that we received during that time interval. And once we have this histogram, we can actually fit a single exponential decay curve to that, and we can extract our lifetime value. And this is shown here from an actual setup in this picture. So on the x-axis you have 12.5 nanoseconds of time, and then on the y-axis you have the number of photons. So number of photons is directly related to intensity, but this is actual number of photons. This is a log scale. The 12.5 nanoseconds is divided into 256 time bins to get better time resolution. And then each of these blue dots is the number of photons that belong to that time bin. Once we have this data, we can actually fit a decay curve to that, or an exponential decay curve to that. Since this is a log scale here, this would be linear. So, and that would give us our lifetime value. And this is just like what error we accumulate in terms of our curve fitting. Okay, moving on. What are the basic components of a TC-SPC-based FLIM system? So here we have our reference pulses coming from a pulsed laser. These go through a discriminator, and then the discriminator actually triggers the start input of a stopwatch. And then down here we have our photomultiplier tube that converts incoming photons into PMT pulses that get amplified and then go through this detector, and then the output of the detector triggers the stop input on the stopwatch. This way we measure the time between the excitation pulse and the arrival of the photon. And then that time value is stored in a memory buffer, and the memory location is dictated by the spatial information that is coming from the scanning system. The stopwatch can be made of either straight time to digital converters, or it can use like analog technique where we charge capacitors and then digitize that data. So why do we, in this picture I showed these CFDs, or constant fraction discriminators. We need these guys because the pulses that we get from the PMT, they're all the same shape, but they're not all the same size. So these are pulses generated from a single photon event. But you can see a pulse could have only this much amplitude, or this much amplitude, or this much amplitude. So then if you use a straight threshold detector, the amplitude jitter in these pulses will be translated to timing jitter, and the lifetime values that we get will not be accurate. On the right hand side here, what we have is where we've used a constant fraction discriminator on the same pulses. So let's say we triggered 20% of the peak of the pulse, so we get the same time for each of those, same arrival time for each of those photon pulses. Once we have a method of determining lifetime at 1 pixel, we can use it in conjunction with the laser scanning microscope. So we can go from 1 pixel to another pixel to another pixel, so kind of scan a longer image and determine lifetime at each pixel. And then we can come up with a lifetime image, which gives us lifetime as a 2D map. So you can see in this image here on the right hand side, this is the fluorescence lifetime image. All pixels here indicate the fluorescence lifetimes at the individual pixels. And in this case, on the left, we throw away the timing information. This is just an intensity image of the same sample, but based on the photon counts. So it's FLIM is pretty easy to integrate into the laser scanning microscopes. Actually, it takes me to my next slide. Generally, the trend has been to couple the FLIM system, or like a third-party FLIM system, to a laser scanning microscope. And what then happens is that your laser scanning software or your multi-photon imaging software is running on one computer, and your FLIM software is running on another computer, which means that your multi-photon imaging data is stored on one computer, and then your FLIM data is stored on the other computer. But what makes things even worse is the fact that your laser scanning software has a lot of capabilities, but we cannot extend those to the FLIM software. What we have done at Bruker is develop our own software, which is our peer review software, and we have integrated the full-fledged FLIM module into that software. So we have one computer that runs peer review software, and we can use that for both multi-photon intensity imaging as well as FLIM acquisitions. The switching between intensity and FLIM is completely transparent to the user. All he has to do is to click a combo box and select the different mode that he wants to use. But the biggest advantage of doing that is that the system features that were available to intensity imaging are now also available to the FLIM mode as well. So our implementation is pretty straightforward and elegant. We already have a femtosecond laser, which is a pulse laser source that you would need for FLIM. We also have detectors on the laser scan microscope. For FLIM, we have to use gas PMTs or hybrid PMTs, and you can use the same gas PMTs for your intensity imaging as well as for your FLIM imaging. We use an SPC 150 card. This is from Becker and Haeckel. They're a pretty well-reputed FLIM hardware software company. We use their SPC 150 card that is housed in the same computer as the other scanning cards. The SPC 150 card offers you several different modes for FLIM. The mode we chose to use is the FIFO mode. And the reason we chose this mode was because we were then not limited by in this mode we can actually stream the data directly from the SPC 150 card into some shared memory location that is accessible by preview. This way we are not limited by the onboard memory on that SPC 150 card. And the advantage of doing that is that we don't have to suffer any loss in either time resolution or spatial resolution or even spectral resolution. The spatial information and pixel clock signals are provided by our own scanning system. The data for FLIM is saved in Becker and Haeckel's SDT format, and the curve fitting and analysis is done offline with Becker and Haeckel's SPC image software. And since the card provides other features as well, if somebody wants to use those features, they can still use them independent of our software. So we don't block those features out. So on this screen, all I'm showing is our preview software. This is our standard software. So here's our control panel, the same control panel that we use for laser scanning microscopy or multi-photon imaging. And here's our standard imaging window. And when we switch to FLIM mode, all we have to do is to go into this acquisition mode and select FLIM from there, and we're ready to do fluorescence lifetime imaging. The standard software gives you the ability to do raster scan images or regions of interest, or Z-stacks, or time-lapse imaging, or allows you to do line scans. It also allows you to do point scans, or as well as tiled images or montages. And the beauty of the software implementation is that all these features are now also available in the FLIM mode as well. So you can be, and these are very useful, especially for example, the region of interest feature. You don't have to scan over the entire field of view. You can just select a single ROI or multiple regions of interest, and then only scan over those, which is very helpful in FLIM because it takes some time to build up the histograms. So you want to be at a pixel for longer to get maximum number of photons. One other dialog that we had to add for FLIM was this dialog down here. This gives us control over the key parameters for FLIM acquisitions. It also gives us a status bar for how different components in the FLIM system are working and what kind of rates we are getting. And then in the end, there is just a screenshot of the analysis software. We use SPC image from Becker and Haeckel. Like I said, we save the data in the SDT format, so we can use this software. There are other softwares that also support SDT format, so feel free to use those if you want. But essentially what we see here, this is the intensity image based on photon counting, And then this is the fluorescence lifetime image. And then this window down here indicates the histogram that we came up with at this pixel where the crosshair is. And then this will give you the lifetime at this particular pixel. The distribution of lifetimes in this entire image can also be seen here. So this is the lifetime, and then this is the number of pixels that have that lifetime. So you can actually see, so if you had like a uniform sample all around, this would be a pretty sharp peak. Yeah, like I said, you can use third-party softwares to do your own or write your own analysis algorithms to do your own analysis for curve fitting and extracting lifetime values. And I think with that, I will actually hand it off to Dr. Scala.
Thank you, Mohammed. That was a really good introduction of FLIM. So we're using fluorescence lifetime imaging to specifically look at metabolism in cells and to look at drug response both in vitro and in vivo. So that's what I'll talk about today. So we've done lots of different cancers, but today I'm going to focus on breast cancer just to keep this kind of focused. And so to understand how we want to implement our changes in the standard of care of breast cancer, it's helpful to first look at the standard of care for breast cancer. So this is first step here is screening, usually mammography. And if something suspicious is found, then a confirmational diagnosis is obtained from a core needle biopsy. And then if indicated, the patient undergoes neoadjuvant chemotherapy in order to shrink their tumor so that they're eligible for surgery. At the time of surgery, the patient is also staged, and adjuvant therapy is assigned if necessary. So the adjuvant therapy consists of three main components. Targeted therapy, which is like the Achilles heel of the cell. We're trying to inhibit specific pathways that drive tumor growth. Also radiation therapy and chemotherapy. There are multiple drugs for breast cancer. Currently, there are greater than 10 standard of care drugs that a patient can be prescribed for their breast cancer care. Usually, we choose about 3 or 4 of these drugs with standard immunohistochemistry. However, 30 to 40% of patients are non-responsive to these therapies, and they're receiving toxicities from these therapies because they are highly toxic. Additionally, there are greater than 50 FDA-approved drugs for breast cancer, so there's a multitude of options, and there's a high toxicity associated with taking the wrong drug. And unfortunately, this resistance is usually determined after months of therapy. So a patient can undergo months of toxicities before they realize that the therapy that they receive is not actually beneficial to shrinking their tumor. So this is the sort of paradigm that we're faced with, and we'd like to streamline this paradigm. So we'd like to determine at the time of diagnosis what is the optimal drug for each patient. Each patient's tumor is different, just as each patient is different. So their treatment should be tailored to their specific tumor. So this is what we're working on. So how are we going to do this? Well, this is our vision. First, we want to take a breast tumor biopsy and apply multiple drug combinations to this biopsy. So we break it up into multiple pieces and grow it into these three-dimensional organoid cultures, which I'll describe later. And this essentially allows us to do a multi-drug test. We can perform optical metabolic imaging. This is a fluorescence lifetime image of NADH, which I'll describe later. And we can use these metabolic readouts to determine the best drug combination for each patient. Once this is determined, we can give that patient their optimal drug regimen in the hopes of increasing the chances of remission-free survival. So this is our sort of pie-in-the-sky idea of how we could help these patients. But as you're probably aware, there are some patients who may not respond to any of the currently available drugs. So the development of new drugs is still vitally important. And you're probably aware of this as well. It's really expensive to develop a new drug, and it takes a really long time. So the estimated cost from identifying a target to launching a drug is about $1.8 billion. It's a lot. It takes about 10 years. And you can see here that the cost is really in these clinical trials, phase 1, 2, and 3 clinical trials. And if you look at where these drugs fail, you see that this is exactly where they fail, in the high-cost clinical trials, especially phase 1 and 2. So ideally, we would want to identify those drugs that will pass through clinical trials at these less expensive preclinical stages so that we can streamline the best drugs for significant research and development in clinical trials and launch. So in order to do this, we need better markers that tell us which drugs are going to be successful or not in the early phases of development. And I argue that functional optical imaging is a really good candidate for achieving this streamlined drug development strategy. So today I'm going to focus on tumor metabolism. So why metabolism is a biomarker for drug response? It turns out that most anti-cancer therapies disrupt cellular metabolism as one of their proximal effects of treatment. And so these metabolic changes precede changes in tumor size. So currently, tumor size is the only standard of care monitoring technique for determining whether a patient is responding or not. So we have this early marker of whether they're responding, so we can determine this at an early time point. I also think it's critically important to look at response on a cellular level because as we're discovering, our tumors are highly heterogeneous and different cell populations respond differently to treatment. So you can imagine different scenarios in which different cells are sensitive to different therapies or different cells are mutated to become resistant to therapies. So in order to identify those subpopulations of poor response, we have to find them on a cellular level, and then we have to identify drugs that work on these highly resistant cell subpopulations because they ultimately are the cells that are driving tumor recurrence, which is what we're trying to avoid. We'd like to just eliminate all malignant cells on the outset. So there are lots of ways to look at cellular metabolism. One that you're probably most familiar with is immunohistochemistry. This is standard. We do this in histology. We look at protein expression. It's great because it's high resolution and molecularly specific. Its limitations are that it's ex vivo, so we can't look at dynamic changes in function of the cells in response to a therapy. And we look at protein expression, which is not really the same as the function of the cell. Patients are currently undergoing some in vivo imaging with FDG PET. This measures essentially glucose uptake through the radioactive analog FDG. And advantages are that it's in vivo, so it's minimally invasive, although you still have to make the contrast agent and inject it into the patient. It requires radio tracers, which are expensive and radioactive. It also, unfortunately, has shown low sensitivity and specificity for determining whether a patient is actually responding to treatment or not. And it's low resolution, so it can't give us that cellular level response to treatment that is critical for determining resistant subpopulations. Another technique under development is diffuse optical tomography, which is great because it's cheap and accessible, and it provides highly specific measures of tumor response to therapy using essentially the hemoglobin oxygenation, water content, and lipid content in the breast tumor. It can be done in vivo and it does not require contrast agents. However, it is low resolution, so it cannot resolve cellular response to treatment. And as I'll talk about later, it's really not amenable to ex vivo high throughput screens, which I think is a good way to measure response to therapy. This is not an exhaustive list. There's many other ways to look at response to therapy, including photoacoustic imaging and MRI. But today I'm going to focus on optical metabolic imaging, which is a measurement of NADH and FAD fluorescence intensities and fluorescence lifetimes. And so these are metabolic cofactors that are already in the cell. So it doesn't require the use of contrast agents, and it provides a holistic measure of cellular metabolism. We've done this both in vivo and in ex vivo three-dimensional organoid cultures. It provides cellular resolution and does not require a contrast agent. However, we know there's no perfect technology, so the limitation here is our penetration depth. So what we've done in this case is balance the ability to do cellular-level resolution imaging with low penetration depth instead of deep penetration depth and low resolution. But we'll see, I think this is an advantage for what we're trying to do. So the endpoints that we're looking at are the redox ratio, which is just the fluorescence intensity of NADH divided by that of FAD. And this is a pretty well-established measure of cellular metabolism. The story is a little complex. So we have, NADH involved in reactions in the cytosol and in the mitochondria, and FAD as well. We also have NADPH, which spectrally and its lifetime also actually overlaps with that of NADH. So we also are sensitive to things like lipid and amino acid synthesis and reactive oxygen species scavenging, reactions in which NADPH are involved. So you can see here when you look at the redox ratio, you have to remind yourself that you're looking at many reactions, and it provides a general idea of a reduction in oxidation state of the mitochondria. Also, depending on the cell you're looking at, it can be modulated by these other reactions in the cytosol. So I wanted to point out where your fluorescent proteins are here. NADH, as I've shown, is involved in lots of reactions in the mitochondria and in the cytosol, and NADPH mostly in the cytosol. So Mohammed already showed this, which is great. You're totally familiar with fluorescence lifetime imaging. I just want to point out special cases for imaging NADH with fluorescence lifetime imaging. We have 2 exponential decay for NADH, and this is due to a quenching of this molecule when it's in its free state. So when it's free, it just self-quenches. It kind of folds onto itself. But when it's protein-bound, it extends itself, and it has a longer lifetime. For FAD, this is actually opposite. It has a short lifetime when it is bound, and it has a long lifetime when it is free. So for both of these molecules, for their biochemical reasons, we fit both of these molecules to a 2 exponential decay so we can resolve their free and protein-bound components. So I talked about the redox ratio, which is just the fluorescence intensity of NADH divided by that of FAD. And then we have the fluorescence decay curves for NADH and FAD, and we can measure this weighted average lifetime for both of them. So if we look at the relationship between these variables, we see some things that are somewhat surprising and somewhat expected. The redox ratio is very well correlated with the glycolytic index, which is just the concentration of the glucose taken up by the cells divided by the lactate secreted by the cells. And this is what we expect based on the oxidation reduction state of the cell and the fact that we produce NADH in glycolysis and consume it in oxidative phosphorylation. This seems like it makes sense. However, if we look at the NADH mean lifetime and the FAD mean lifetime, we see that these are not correlated with the redox ratio. So they provide added value in the molecular microenvironment of these molecules. So the redox ratio provides information on the glycolysis levels and the oxidative phosphorylation levels in a cell. And the NADH mean lifetime and the FAD mean lifetime provide information on protein binding, activity of enzymes and such. And we know that both of these variables from previous papers are very sensitive to cellular metabolism, but in different ways than their intensities. So it makes sense to put all of these variables together because they provide added value when we put them together. And I'll show you data that supports this later. So what we've done is a multivariate analysis of variance to determine how to combine all of these very important metabolic variables. And what we've done is just a linear combination of the redox ratio, NADH mean lifetime, and FAD mean lifetime. This minus sign here just comes down to the fact that FAD has a short lifetime when it is bound to a protein, whereas NADH has a short lifetime when it is free in solution. So what it provides us is a conglomerate variable that tells us about relative glycolytic levels and the metabolic microenvironment of these molecules or their protein binding. And we empirically use this in the data that I'll show you next. So this tells us about the relative amounts of NADH and FAD and the enzyme activity of those molecules. In our drug assays, we have designed this OMI index to increase in cells that are resistant to therapy and to decrease in cells that are responsive to therapy. So decrease in the OMI index indicates that that patient will respond to that drug treatment. So Mohammed also went over the instrumentation quite well. I just want to tell you what we used for excitation wavelength, 750 nanometers for NADH, 890 nanometers for FAD. Remember, these are two-photon excitation wavelengths. I've shown their emission spectra here, but remember that NADH and FAD have very different excitation spectra. So we've shown with specific metabolic perturbations that we are able to specifically look at NADH and FAD contributions. We also are interested in cellular level analysis, so we have modified some freeware available. I encourage you to look at Cell Profiler. It's great freeware and you can modify it as necessary. We modified this in MATLAB. because we specifically want to look at the cytoplasms of cells. We developed a segmentation routine to specifically segment the cytoplasms of the cells. We extract all 8 of our variables, including NADH and FAD intensities and their lifetime components, as well as combination variables such as the redox ratio and the weighted average lifetimes for NADH and FAD. Since we're interested in cellular level response to therapy, we also looked at the distributions of these variables by fitting them to multiple Gaussians and choosing the best fits using the Akaki information criteria. That's what AIC stands for. So for our in vivo studies, we were specifically looking at trastuzumab, which is also the other name for this drug is Herceptin. It's a HER2 inhibitor given to about 20% of breast cancer patients that overexpressed HER2. However, 30 to 40% of these patients exhibit de novo resistance to this drug, so that means they never respond to this therapy, yet they're subject to the toxicities of the therapy. So we wanted to look at some clinically relevant xenograft models, which include HER2-positive human breast cancer cells that are responsive, that's the BT474 cells, or resistant to trastuzumab, because this mimics the clinical presentation of resistance. So we just took these cells and implanted them into different xenograft mice models. So first we wanted to look at the tumor size as a function of time, and as we expect, if we don't treat these tumors, they continue to grow over the time course, and these Herceptin responsive tumors shrink over the time course. So as I mentioned before, FDG PET is a standard in vivo measurement for measuring tumor response in breast cancer patients, but it is subject to some difficulties with sensitivity. So this is an FDG PET image, and the T's here are pointing to the tumors. So we looked at the amount of FDG uptake in these tumors for the control and trastuzumab-treated tumors over time. And in contrast to the tumor growth curves, we don't see any changes in FDG uptake between control and trastuzumab-treated tumors. We also performed in vivo fluorescence lifetime imaging of these tumors. So we placed, the living, breathing mouse on the microscope stage and put the objective up there in the mammary gland. These are orthotopic tumors, so they're grown in the mammary gland. And we imaged the cellular level response to treatment using our metabolic imaging techniques. So this is a representative in vivo image. And this is our OMI index as a function of time in these animals for the control and the treated tumors. So you can see as early as day 2 after one trastuzumab treatment, we see a significant change in the metabolism of these treated tumors compared to control. So we also wanted to look at a more clinically relevant model, which is the Trastuzumab-Resistant model. And as expected, these are resistant tumors, so they grow in the presence of drug, and they also grow without drug. So they pretty much look the same whether they're treated or not. And we did the same comparison with FDG PET imaging. These are your tumors here in the mammary gland. And over time here, we saw actually an increase in FDG uptake in the control tumor between 2 days and 12 days of treatment, but no changes between the control and treated tumors. This is another in vivo image from the mammary gland of a living mouse, and we see as expected here that our OMI index does not differ between treated and control tumors over the entire time course. So this indicates that we can resolve not only response but a lack of response due to the changes in the fluorescence lifetime of NADH and FAD. And finally, we looked at a partially responsive tumor model. So this model is known to have about a 70% response in tumors, and about 30% of the cells do not respond. So we looked at the cellular level response in these tumors. This is the control in blue and the treated in red. And we see again that about 70% of the cells responded, and about 30% of the cells did not respond to treatment. Again, this was done in vivo in xenografts that are grown from MDA MB361, partially responsive tumors. So those are the in vivo studies, but now I want to talk some more about the high throughput screens, because these have particularly important translational implications. So primary tumor organoids are great because they can include cancer cells as well as native stromal components, so we can enable communication between cell types, and this includes fibroblasts and immune cells. We know that 3D culture more closely mimics in vivo-like conditions because it replicates microenvironmental gradients of oxygen, glucose, and pH. So we want to use our fluorescence lifetime imaging to identify subpopulations of response within these intact organoids over time. And we actually did this in a few patients, and I will show you those results. And we're moving on actually to validating this in patients. So for these studies, we just took the same two tumor models, trastuzumab responsive and resistant xenografts. We removed the tumors and immediately digest them and minced them, and then embedded them in Matrigel so that they could grow in 3 dimensions. We imaged at 24, 48, and 72 hours post-treatment with our Two-Photon FLIM system, and we measured response to clinically relevant drugs, including paclitaxel, which is the standard of care chemotherapy, tamoxifen, which is an anti-estrogen receptor therapy, trastuzumab, which again is the anti-HER2 therapy, and XL147, which is in clinical trials right now to be used in combination with trastuzumab in patients who have developed a resistance to this drug. So in our responsive xenografts, we see, as expected, when these tumors are not treated, they grow, and they actually respond to all of the treatments that we gave them. This table here just shows the day at which we first resolved a significant change in tumor size. So these are all the in vivo validation data that we are then going to compare to our ex vivo organoid response. So this is just a Two-Photon FLIM image of NADH in our organoid. And then at 72 hours post-treatment, we measure multi-drug response to not only the individual drugs, but their combinations. So as we see, as in the in vivo studies, in the organoids, we resolve response to all drugs and no response in control tumors. However, we can resolve this response on the cellular level, and you can see that there are subpopulations of varying response to this treatment. We also validated with standard immunohistochemistry. This is cleaved caspase-3, which is a measure of apoptosis. And we also find that there is increased levels of apoptosis in all of the treated experimental groups. So I think the more interesting comparison is that in trastuzumab-resistant tumors. So these show a much more varied response to treatment. So the trastuzumab-treated tumors are very similar to that of control tumors in terms of their tumor growth. And then we have these 2 other treatments, paclitaxel and XL147. These tumors initially shrank and then grew. So this is very relevant to the clinical context of a patient who initially responds and then relapses after extended treatment. And then with the combination treatments, we see that all of these tumors continued to shrink in the presence of these drugs. The question now is whether we can replicate this more complex response in organoid culture. And this is a representative organoid image from the fluorescence lifetime image of NADH in an HR6 xenograft. So it's important to look at the time course of the treatment is what we discovered in this experiment. So initially we see a response in the paclitaxel and XL147 treated groups and no response in the trastuzumab group. But then at 72 hours post-treatment, these tumors regress so that they show an increase in the OMI index at 72 hours post-treatment. So this is mirroring the response and regression that we see in the tumor growth curves, but with the OMI index at 24 hours and 72 hours post-treatment. In the combination treated tumors, we see response both at 24 hours and at 72 hours, indicating sustained response. This again agrees with standard measures of apoptosis in the organoids at 72 hours post-treatment, which again show sustained response in the multi-drug treatment groups. So I wanted to show you a specific example of how cellular level resolution can tell us a little bit about the subpopulations and how they're responding to treatment in these more complex tumors. So at 24 hours and 72 hours, we see that the trastuzumab-treated and control tumors are essentially overlapping in their cellular response to treatment. If we then look at the paclitaxel-treated group, we see an initial response at 24 hours and then a regression at 72 hours. So then this cell population is again overlapping with that of control and trastuzumab by 72 hours. However, if you look at a combination treatment group, this is all 3 drugs together, we see an initial bimodal response, and then at 72 hours, we see that this bimodal response is becoming more unimodal and less overlapping with the non-responsive groups. So this is an interesting way to monitor the response of cellular subpopulations to see how they change with sustained treatment. I just wanted to show a little bit about how we've established feasibility for using these methods in patient tumors. So far, we've looked at 6 patients. We have additional patients I don't have in this talk, but I wanted to give you an overview of triple-negative breast cancer patients, ER-positive patients, and HER2-positive patients. And this is sort of representative images of the fluorescence intensity of NADH and FAD, the redox ratio, the mean NADH fluorescence lifetime, and the mean FAD fluorescence lifetime. So these tumors were obtained from patients at the time of surgery, and then we digested them and embedded them in 3D Matrigel into these organoids. So we found in immortalized cell lines earlier in this investigation that we could differentiate tumors by their receptor status based on the metabolism of those tumors. And we found that we could also do this in primary tumor organoids that we obtained from patients. So if we grow these organoids from a patient with triple-negative, ER-positive, or HER2-positive breast cancer, we can differentiate these subtypes of breast cancer by their OMI index. If we look at response in one example patient, which is the HER2 or ER-positive patient, we see varied response. So this patient in particular was a ER-positive, HER2-negative patient, so they did not respond to the antibody against HER2 as expected, but this patient did respond to all other drugs alone and in combination. And if we look at their cellular level response to treatment, we can see that there is a heterogeneous response even in the multi-drug treatment groups. And in general, we see more heterogeneous response in our human patient tumors than in our xenograft-derived tumor organoids. And this makes sense based on what we know about human tumors and xenograft tumors. So we were able to detect drug-induced changes in human organoids, and the single-cell analysis allowed for the identification of cellular subpopulations of varying metabolism. And these population curves are broader for human versus xenograft tumors, which is what we expect. So for validation so far, we have standard measures of apoptosis, which is cleaved caspase 3, and we see that these standard measures agree with our metabolic measures of response in organoids. So these are just some examples of different patients who were also estrogen receptor positive patients, just to show that there's varied response to treatment as we expect in the patients. Not all of them do respond to the ER inhibitor, even though they are ER positive tumors. And we are investigating validating this right now with respect to neoadjuvant therapy response at the time of surgery. So from the studies, they were published by the same author, but they're published in 2 different years in cancer research. The in vivo studies were published in 2013, and all these organoid studies, including the human data, was published just a month ago. So OMI detects early molecular changes in organoids that can streamline drug development. It allows for faster readouts and fewer animals to be used to measure drug response, because these organoids require only one tumor to measure response to multiple drugs in combinations. Single-cell analysis reveals tumor heterogeneity and could identify subpopulations of drug response and resistance in vivo, which is really important for developing drugs. And we believe that this high-throughput organoid OMI screen could predict optimal drugs for individual patients, and we're validating this in the neoadjuvant setting right now for breast cancer. So I'd just like to thank all of my wonderful collaborators here at Vanderbilt, Rebecca Cook, Carlos Arteaga, and Alex Walsh, who is a graduate student who did all of this work, and of course my funding. And with that, I think we are going to talk about questions.
Yeah, great. Thanks a lot, Melissa. That's a tremendous presentation. So we'd like to open things up to questions. So for the folks attending the webinar, if you have any questions, you can type them into the question area on the interface for the webinar. Okay, so there's no questions. I'd like to thank you for your attendance today, and